Hello everyone, and welcome to this podcast for cell biology. In this podcast, we're going to talk about the topic of protein folding, which we started on the last podcast. But primarily what we want to talk about in this podcast are two big items. The first is, how do proteins fold? And then the second will be a short discussion on protein function. Let's go ahead and get started with how proteins fold. Later on in the semester, we're going to introduce the laws of thermodynamics. But I think we need to at least mention briefly the second law of thermodynamics, because it plays a very important role in how proteins fold. A very generic definition of the second law of thermodynamics is that systems tend to go from an ordered system to an unordered system. Another way of saying that is that systems increase in entropy. But for now, what I really want you to know is that systems that are ordered are constantly fighting to stay ordered because their natural tendency is to become unordered. So if we think about that in the context of a protein, let's first draw our primary structure, where we have our N-terminus here and our C-terminus here. It moves to a secondary structure, or I should say it folds into a secondary structure, where we will have maybe an alpha helix, maybe a beta sheet, so a little bit more order. Then next, this secondary structure begins to fold upon itself, and it forms an elaborate tertiary structure, still with the N and C terminus, but again, more order. This may, but it doesn't have to, as you may remember, fold into a quaternary structure where two of these subunits or two different subunits interact with each other, in which case we have, again, even more order. As we'll learn in a little bit, that this process of going from primary to tertiary and quaternary is spontaneous. It's hard to imagine going from a very unordered structure to a very ordered structure, which we know because of the second law of thermodynamics should require a lot of energy. How could it possibly be spontaneous? So how does it do this? Well, first, let's consider two factors that are important in driving this spontaneous reaction. The first is the primary sequence. Remember, the linear sequence of the amino acids in the primary structure ultimately determine how the atoms are going to interact in order to form the tertiary and the quaternary structure. But this other point is the one I want to focus on, and that is energy. In order to go from this unordered structure to a very ordered structure, we said before, requires lots of energy. Where does that energy come from? It turns out that energy comes from the formation of chemical bonds. So the hydrogen bonds that help form the secondary structure that we've talked about before, the hydrogen bonds, the ionic bonds, the van der Waals, the non-covalent or hydrophobic interactions, all of these spontaneously form because of the primary structure. And when they spontaneously form, they release energy. And that energy is what helps drive the spontaneous formation of these very highly ordered proteins. And maintaining this order requires that excess energy. Okay, I've now just told you that this process is spontaneous, but we didn't always know it was spontaneous. So let's talk about how we figured that out on the next whiteboard. As scientists began to study protein folding, one of the big questions that popped up was, can proteins fold spontaneously? Now there was this one scientist who did a very elegant experiment. His name is Anfinsen. You don't need to remember his name, but he was the one that did this experiment. It's a really important experiment. He took an enzyme called RNase A, and so let me just draw it like so. And this RNase A enzyme was stabilized by various disulfide bonds, as well as some other forces such as hydrogen bonds and electrostatic interactions. I should also mention that RNase A, its job is to degrade RNA. He took this enzyme and added a reducing agent to beta-mercaptoethanol and urea, and that helped break these disulfide bonds. And what happened then was the protein unfolded, and it's also non-functional at this state. What he discovered was when he removed these chemicals, so no 2-beta-mercaptoethanol or urea, unfolded protein here would fold back and be functional. This suggested that this protein, this enzyme, could fold spontaneously, and indeed it can fold spontaneously. Now it's important to note though that even though Atkinson showed that this enzyme and others showed that other enzymes can fold spontaneously, we now know that the folding of the protein often requires helper proteins. So let's talk about those next. So to answer this question, can proteins fold spontaneously? Let's just write sometimes. I just mentioned that RNase A could fold spontaneously without the help of other proteins, but sometimes there are other proteins which require help. And there's a whole family of proteins called chaperones, and chaperones assist in the folding of some proteins. Really, I should probably say most proteins, because most proteins do seem to require some help here. And even the ones that can fold spontaneously, we often find in physiological conditions, they often will use chaperone type proteins. So what I'm going to do here is I'm going to draw a ribosome, as lovely as it is here. And this ribosome will have a messenger RNA being fed through it. And as this messenger RNA is being translated into protein, we see protein coming off the side here. Chaperone proteins will often just immediately bind to these other proteins and help fold them. They will position the growing amino acid chain here, the growing protein chain here, in such a way that encourages the right folding, the correct folding of that protein. So as this protein is coming off here, these chaperone proteins bind to specific sites there and help assist in the final folding of this protein. And then the chaperones are released to go back here and redo the job with new proteins being made. In other instances, you could have a chaperone that acts like a chamber. I'll draw it like so. Proteins that are being made before they get folded, and before they get folded incorrectly, are taken inside of this chamber. And inside of this chamber, they're locked in here, so to speak. And in the chamber, because the area is much smaller, it encourages the right folding of this protein. So eventually it will fold in a correct form and then be released from this chaperone protein. Okay, now let's talk about this next topic of the importance of protein flexibility. So on the last slide, we talked about how we would take this primary structure protein and how that was folded up into a secondary structure and then a tertiary structure and then sometimes a quaternary structure, but I'm just going to stop with a tertiary structure here. And even though energetically this is a spontaneous reaction, there are still often chaperones involved in folding this tertiary structure. However, this tertiary structure, at least initially, is often inactive. Very elaborate mechanisms are in place to make sure it forms in this way, but often it is inactive, as I just said. But it has the flexibility to become active, to fold in a slightly different way that makes it active. Still a tertiary structure, but now it's active. Why is that flexibility important? Why not just let the chaperones make a fully active protein? Why do we have to have this additional step? And What allows it to do that? What causes the activation of this protein? As we consider that question, it's important to remember that protein structure is equal to protein function. It has to fold in this very precise way here to be functional. It can fold in other ways, but it's not functional. And so with that in mind, we should recognize that we don't want all proteins to be functional all the time. So maybe this protein here is the protein insulin. We only want insulin to be active at certain times. So we wouldn't want it to be in this active folded state all the time. We would want to have a way to turn it on when we need to. And so that's the main point of this comment here. We don't want it to be functional all the time. However, we want many of our proteins, not necessarily all, but many of our proteins to be poised to act. While we don't want insulin to always be active, when we get done eating a meal, we want it to be ready to act. So it's much quicker and much more efficient to go from this inactive, poised state to being an active protein. Much quicker to do it that way than to go all the way back here to the gene that's not even shown on this board and create the protein and then fold it every time you eat a meal. It's much more efficient to have this inactive protein present and then to activate it in just one step once you eat a meal. And insulin was just an example. Many of our enzymes function this way. We want them to be inactive until we need them. It could be destructive to have them active all the time. So we answered why it's important, but how does it do it? How do we allow this protein, this inactive protein, to become active? So let's talk about that quickly here. We can think of two ways to activate an inactive protein. The first is that the inactive protein could interact, or you might say bind to, various ions or molecules. Some ions it might bind to could include calcium, magnesium, maybe manganese, various ions that when they bind to this inactive protein, they could alter the charge of this protein and allow it to fold in the active form. So this is a very common way to activate a protein. Another common way is adding certain kinds of molecules. You might add a methyl group. A phosphate group is a very popular way to cause these proteins to fold and become active. And there are some additional ones too, but just talk about those two for now. Another way is that the inactive protein could be modified. So for instance, maybe we had a protein that was shaped like this. And for it to become active, part of the protein had to be removed. Or we might say it was cleaved. And now the protein looks like that. And it is now active. So two ways to activate a protein. You could add an ion or a molecule to an inactive protein, or you may have to modify it in such a way like removing part of the protein to make it functional. There are many examples of both of these within a cell. Both are very common, and in some cases, both are used to activate a protein. Next, I would like to talk about how folding or protein folding can be infectious. And in explaining this, the best example are these structures called prions. Some example of prion diseases include something called Creutzfeldt-Jakob disease, and we'll just call it CJD. And these affect humans. There's also bovine spongiform encephalopathy, and we'll just call that BSE. And you probably popularly know this as mad cow disease. There's also chronic wasting disease. I'll just put CWD. This is what affects deers. And there are often a few cases every year in Michigan of deers affected with chronic wasting disease. All of these diseases affect the brain and make holes in the brain and will lead to death. In fact, some of these are transferable, so humans can catch these prion diseases from cows who have BSE or deers that have CWD, particularly if you consume any part of the brain. And the reason for that is because these prions are infectious. It's not an infection caused by bacteria or viruses or fungal pathogens. These are proteins that naturally exist in our brains that can be converted to an incorrectly folded protein. So these diseases are caused by a protein in our brains, and these proteins are called PRP, which just simply stands for prion protein. Though I'm not going to draw this structure completely accurate, the normal PRP will have a series of alpha helices. And so now this normally folded PRP protein can do its job. And we'll talk more about this in class, but what I want you to know for now is that the normal PRP forms these alpha helices, predominantly alpha helices. Now, every now and then, this PRP protein can fold incorrectly. And while you may still have one or two alpha helices, the predominant secondary structure in this incorrectly folded protein are beta-pleated sheets. And so this is the misfolded PRP. And this is the one that can cause problems in the brains. Can lead to an altered function, can lead to forming those large areas that appear as holes in the brain, that are holes in the brain. Now, if it was just this protein that misfolded every now and then, it wouldn't be a problem, because proteins misfold all the time, and they're just degraded. But for some very interesting reasons, this misfolded PRP, the one that can cause the diseases, is infectious, meaning it can interact with the normally folded, the correctly folded PRP proteins, and convert them into the predominantly beta-pleated sheet form. And in doing so, it makes these normal or correctly folded PRP proteins now misfolded and capable of causing the disease. And so in this way, this one or two misfolded proteins can go through the system and cause the properly folded proteins to misfold. And as I said in class, we'll spend a little bit more time talking about this particular disease, because it's something that you'll come across probably at some point in your academic career here, or maybe in your profession. Okay, now let's talk about protein function, and we're going to focus a little bit more on one of their functions called catalysis. In your note-taking guide, I list several functions of proteins, including functioning as a catalyst, functioning in defense, movement, signaling, structure, transport, and that's it. You can read the book to get a little bit more information about each of these, and we'll talk about all of these throughout the semester, but just a real brief few words here. Catalysts, these are what we might think of as enzymes. They speed up reactions. Defense, we can think of several examples of this, but primarily let's think about the immune system, and in particular, antibodies. Antibodies are proteins that help fight off infection. We can think of moving as movement of the whole cell, or intracellular movement. Things within the cell moving. Signaling. I'm going to draw this one. There are proteins that are stuck on the outside of a cell that can receive some kind of a signal. And when they receive that signal, they cause something to happen within the cell, often by modifying other proteins in the cell. So signaling proteins play a lot of different roles outside the cell and in the cell, but this is a key way that the cell senses the environment, and it's important in daily life of a cell, but also in things like the death of a cell and the development of a cell. Structure. As the name implies, it provides structure to the cell. Things like our skeleton is made up of structural proteins. Our hair, fingernails, are all examples of structural proteins. Transport. Within a cell, we will have often these transport proteins that bring chemicals from outside the cell into the cell, or sometimes we're moving water into the cell. But these are all transport proteins. But let's spend a little bit more time with catalysts. We'll learn throughout the semester that catalysis can be caused by proteins or RNA molecules. But primarily, most catalysts in a cell are proteins. So why do proteins make the best catalysts? And the reason for this, why proteins are the best catalysts, has to do with the fact that we talked about before, that they are flexible. An enzyme, a protein enzyme, maybe is responsible for binding two substrates. For simplicity, I'll just draw a square and a circle. And the job of this enzyme is to take those two substrates and make one product by linking them together. These two substrates are rarely going to just spontaneously do this without the help of an enzyme. And what enzymes will do then is they'll bind these two substrates, and in the process of binding these substrates, the enzyme, the protein enzyme, will shift its shape and bring them into close proximity to each other, allowing an increased chance that the bonds will form between these two substrates to produce the actual product that we want here. So because proteins have this flexibility and can bind these substrates very precisely and then bring them together in a particular orientation to facilitate the proper formation of the product, proteins make excellent catalysts. In the past few podcasts, we talked about all the really great things that proteins can do. The functions they have, the way they can change shape, the way they help cells perform a diverse and wide array of functions. So did life arise from proteins? Did proteins come first, so to speak? And the answer to this question is probably no. And the main reason for that is that whatever this first molecule of life was, if it happened to have been proteins, there would have to have been a way for this one protein to replicate itself and make two identical proteins. With every time a cell divided, it would have to make an exact copy of it. There's no evidence in current life or in past life that proteins have the capacity to do this. DNA, as we'll talk about in our next podcast, has the capacity to replicate itself. So is DNA the first molecule of life? And the answer is probably also no. Let me put no here, and no by DNA. And the main reason it's probably not the molecule of life is that DNA, even though it's a great hereditary information and it codes for everything that we need, it itself has no catalytic ability. That first molecule had to have a way to copy itself, but also had to have a way to function by itself. So let's talk about our last choice here, and that's RNA. RNA can act as a catalyst, like proteins. Not quite as good. Proteins would eventually take over the role of the catalytic functions of RNA, but they can act as a catalyst. They can also act as hereditary molecules. They can copy themselves. Though we don't know for sure, it does seem like the RNA molecule is most likely the molecule that life arose from. And over the many millions of years, some of the functions of the RNA molecule have been replaced. Proteins are better catalysts. DNA is a better hereditary molecule. But RNA still plays many functions, valuable functions in the cell. Okay, that's all I have for this particular podcast. But it's just a real quick summary. So we talked about protein folding. We talked about how it was spontaneous, at least energetically spontaneous, but it did often, or does often, require molecules like chaperones. We also talked about how it was important that that protein, once it was folded, remained flexible. And we talked about the infectious nature of some proteins, how they could cause the misfolding of other proteins. And then we closed with a discussion on protein function and what is likely the first molecule of life. And we said that the best candidate for right now is RNA. Okay, that's all I have. But if you have any questions at all, please make sure you come see me. If not, I'll see you in class. Bye.